Breast cancer is the most commonly diagnosed cancer, with metastasis being the primary cause of most breast cancer-related deaths. 1.6 million cases of breast cancer are diagnosed annually, with severity ranging from early, curable stages to metastatic breast cancer. In metastasis, cells detach from the extracellular matrix, or the ECM, to move to a secondary site. First, cells break away from the primary tumor. After invading surrounding tissue and traveling through the bloodstream, they form a secondary tumor. Normal cells undergo anoicus upon detachment, or programmed cell death. However, ECM-detached cancer cells survive detachment in part because they maintain cellular energy metabolism. In normal cells, glucose is used for energy production to support energy needs. Unlike normal cells, however, cancer cells undergo the Warburg effect, where glucose is redirected away from the TCA cycle. To survive, cancer cells may use the amino acid glutamine as an alternative energy source to replenish the TCA cycle through the TCA cycle intermediate alpha-ketoglutarase. Two cell lines were used in these experiments. The first is an early model of human breast cancer, the MCF10A RAS cells, which I will from now on just refer to as RAS cells. The second cell line we used is a human metastatic cancer line, or MCF10CA1A cells, which I will now refer to as just 1A cells. The purpose of this study was to compare the differences in % decrease of cell viability during ECM detachment using RAS and 1A cells in glutamine-depleted conditions. We also measured gene expression for enzymes involved in glutamine metabolism in attached and detached RAS cells. While the effect of glutamine in ECM-detached conditions in metastatic cells is not known, we hypothesize that ECM-detached RAS cells will have a greater decrease in viability in glutamine-deprived conditions in the 1A cells. In ECM-detached conditions, the RAS cells had a 58.4% decrease in viability, while the 1A cells had a decrease in viability of 32.7%. In glutamine-deprived conditions, the opposite was observed. The 1A cells had a 15.2% greater decrease in viability than the RAS cells. The greater effect of glutamine depletion on the viability of the metastatic cells suggests greater dependence on glutamine metabolism in ECM-detached conditions. We also measured mRNA expression of two genes involved in glutamine metabolism. The first one was the glutamine metabolizing enzyme, glutamate dehydrogenase. In detached conditions, mRNA expression was decreased in the non-metastatic RAS cells when compared to attached conditions. Additionally, the glutamine synthesizing enzyme, glutamine synthetase, showed increased mRNA expression in detached conditions in non-metastatic cancer cells, suggesting that in ECM-detached conditions, the RAS cells decrease externally sourced glutamine metabolism and switch to synthesizing glutamine. From this, we concluded that the metastatic 1A cells are more susceptible to decreased viability in detached glutamine-deprived conditions compared to non-metastatic RAS cells. Since we only looked at genes involved in glutamine metabolism in the RAS cells in this experiment, one future area of study may be measuring glutamine metabolizing enzymes mRNA expression in the attached and detached 1A cells. Additionally, we plan to measure cell viability in glucose-deprived conditions for the 1A and RAS cells. A comparison of metastatic and non-metastatic breast cancer cells will provide insights to direct the development of new targets for the prevention of breast cancer metastasis.